Thank you, Dr. Henningfield. And we welcome Dr. McCurdy, who is a professor at the Departments of Medicinal Chemistry and Pharmaceutics at the University of Florida. He's the director of the Translational Drug Development Corps. Dr. McCurdy is a broadly trained pharmaceutical scientist and pharmacist whose research has focused on the design, synthesis, and development of drugs to treat pain, anxiety, and substance abuse. He is an internationally recognized expert on Kratom. He served as the president of the American Association of Pharmaceutical Scientists and serves as a consultant to the U.S. Food and Drug Administration's Drug Safety and Risk Management Advisory Committee. We welcome Dr. McCurdy to this discussion.
Thank you, Mac, for inviting me. Thank you for hosting this event to the United Nations Commissions on Narcotic Drugs. It's really a pleasure to be here and to share a lot of our work with you. I can tell you one thing, we all agree on the science that we put out. We can't agree on how to pronounce this thing. So we call it Kratom, Kratom. It depends on where you are in the world. It's all tomatoes. We can't even agree on the alkaloid pronunciations either, but we'll go from there and we'll try to make it all make sense. So what we've talked about really up to this point is the use in the United States. And I want to say that use in the United States looks something like this. It's widely available across the internet. It's available in smoke and vape shops. And the American Kratom Association reported back in 2019, which I'm sure this number is greater now, that almost 2,000 metric tons were exported to the US every month. If we do just some quick back of the napkin calculations with a typical dose, which has been discussed, around 3 to 5 grams, this suggests that there's over 15 million users in the United States at this time. Now, there have been some documented reports and surveys that show that number could be somewhere around 2 to 3 million, but based on sales and product movement, it definitely appears to be greater than 15 million users currently in the United States alone. Those products definitely look like what you see in this picture. They're ground leaf, powdered leaf materials. They are encapsulated into capsules. Some are pressed into tablets. Others are made into extracts or liquid shot energy drinks. This is what the product like in the US. East Asia, it looks very different. So the traditional use was, again, for field workers to relieve pain, as a stimulant to improve their work capacity, improve their mood, and also to reduce opioid withdrawal. And this has been documented that many of these workers that would use opium, not prescription opiates, when they would run out of opium, they could forego withdrawals by increasing their kratom consumption. And so what you see down in the bottom is a picture of a tree in a flowering stage. Up in the top right-hand corner, you see those leaves that are picked fresh off of the tree, ripped or cut and put into boiling water. So this is a very different product than what we see with dried leaf material that comes into the United States. That material then is packaged into some sort of baggie or vessel that can then be used throughout the day. And in general, these individuals say that the juice that they call it, or this tea or decoction, lasts for about 24 to hours, and then the efficacy in their mind is gone. Recently, this was pointed out by Dr. Henningfield, we've shown that polydrug users, those that are using opioids and methamphetamine, are using kratom to reduce that. And the predominant active ingredient we've already discussed is mitragynine or mitragynine, depending on how you want to speak about it. The therapeutic potential is really great, we believe, for this. And we've shown this through numerous studies, not only from our lab, but other labs as well. And we need to do a lot more work because most of the work that's out there right now is in acute settings. So very acute studies, even in the human studies that have been done, I wouldn't consider these to be chronic dosage studies, not that you would see in something like opioid detoxification. But we believe that kratom has the potential to replace several medications used during the opioid detoxification process, and that alone would help improve medication adherence and chances of individuals completing detoxification. We also feel it could be a good medication-assisted therapy. We've already heard that there's low subjective opioid withdrawal scale scores, and that polydrug users are reporting kratom use to reduce methamphetamine use. And then all of this has been sort of blocked by a lack of standardized product that has really prevented rigorous clinical trials. And these are actually starting to come underway. And as we heard, some of the results are about ready to come out in full force. We decided to take things in our own hands at the University of Florida, and we have a quite large group in our horticulture program that is learning how to cultivate these trees. We're growing these trees, we're studying them genetically, we're understanding how these trees are producing the alkaloid pathways. We've just started to publish on some of the alkaloid biosynthesis and understanding of that. And this is a snapshot of some of the 40 plus alkaloids that have been isolated from kratom. And what I wanted to emphasize here is that we've heard a lot of talk about mitragynine or mitragynine and 7-hydroxygynin. However, these are only two pieces of a very complex symphony orchestra that I like to call it, where we have multiple instruments playing at the same time, making some sort of music that is totally different than when we pull one of those instruments out and listen to it at full blast. And so you've heard about some of the full blast studies. I'll show you a little bit more of full blast studies as well. But many of these compounds are also just isomers of each other. So left-handed and right-handed versions or diastereomers of each other. And to make that as simple as possible, they're basically mirror images of each other. And so they have the exact same chemical constituency or makeup, but they have completely different pharmacological effects. And I want to underscore how complex this is. Where we talk about opioid, we talk about adrenergic, and indeed, mitragynine itself has partial agonist activity at the mu-opioid receptor, which we know is biased in its signaling and not recruiting beta-arrestin, and we've heard this before, which is thought to be one of the key and fundamental facts about it being a little bit safer. But we tend to feel that its safety is really related to its polypharmacology and its complex pharmacology, and not the fact that it's a pure opioid. And I want to try to stress on everyone that it's not a pure opioid. It is interacting with opioid receptors, but it's interacting with alpha adrenergic receptors. It's also interacting with serotonergic receptors, and I'll show that to you in a little bit. One thing that we note is a critical review of Poison Control Center data in the United States shows that kratom overdoses resemble stimulant overdose, not opioid overdose. This is a huge distinction. We're not seeing people having respiratory depressive events. We're seeing people that are becoming agitated, cardiovascular increases, and also seizures, as many of the overdose, if you will, or too high exposures that Poison Control Center emergency department visits. As for 7-hydroxymitragynine, this is a metabolite that's generated from mitragynine in our bodies. And this does have very high affinity for mu-opioid receptors, and it is a potent mu-opioid agonist. And there's been a lot of concern and discussion that this molecule in and of itself is what's responsible for the analgesic effects. I'll address that in just a moment, but I want to just say a few take-home messages about mitragynine. And again, this is mitragynine as the single alkaloid, not as kratom. It is a polypharmacological alkaloid with at least a mix of opioid, adrenergic, and serotonergic pharmacology. It shares some effects with mu-opioid agonists, but not all. It has very low potency with very low mu efficacy, which is really a hallmark for what you want in a drug treatment for opioid use disorder. Now, I'm going to say that I could have spent probably a whole day with you all talking about the data we have, but what I'm not going to show you here, but it's available in our manuscripts, is that we see less tolerance development than morphine at equal analgesic doses. We see less dependence than morphine. We also see discriminative stimulants effects that are very different from morphine. Dr. Henningfield mentioned that we see a discriminative stimulus cue towards alpha adrenergic molecules like lofexidine or clonidine than to opioid molecules like morphine. We also see less tolerance, less abuse, less dependence liability than other opioid analgesics. And we do not feel that mitragynine alone has abuse potential or addiction potential, at least in animal models. We have seen that it will reduce morphine intake and heroin intake in rats. And again, these are desired characteristics of a candidate pharmacotherapy for opioid addiction and withdrawal. The take-home message about 7-hydroxy, which is the metabolite in minor amounts, and one that we've talked about, really putting some standards in place in products, which should be considered a kratom constituent with high abuse potential and may increase the intake of other opiates. And again, this is as the molecule alone, not in complex with the entire natural product. It's a very selective opioid receptor ligand. We haven't seen a compound this selective for opioid receptors before. However, the extent that mitragynine is converted to 7-hydroxymitragynine in our bodies appears to be insignificant in the terms of the pharmacological ramifications, and we've published on that as well. So I want to jump out of the individual alkaloids a little bit and into the full plant material. And what we see here is, in comparison to morphine, LKT, which we call lyophilized kratom tea, where we've made the tea in a traditional sense, and then we freeze-dried or lyophilized that. I also call it astronaut tea. But we can see that it clearly has much higher doses that are needed to achieve the analgesic effects of morphine. It never approaches 100% analgesic efficacy. And you can see that even at 1, 2, and 4 grams per kilogram, we sort of hit a ceiling effect with the analgesic efficacy of kratom. So it almost has its self-regulating piece within that tea in those doses can last for quite a long time. And this is in the mouse. And so what we're seeing is almost an hour and a half worth of analgesic relief in a mouse, which is translatable to quite a significant longer time in a human. We also have done some pharmacokinetic profiling of the whole plant material to understand what alkaloids are actually being exposed in the blood supply from that complex symphony orchestra of almost 40 plus alkaloids. And what we've seen really is that mitragynine, the major alkaloid, is there. We also see speciosialatin in high levels. We haven't talked about speciosialatin or what it does. We see 7-hydroxymitragynine, which actually is created from metabolism. If you look at the bottom line here of the lyophilized kratom tea, it's below our limit of quantification, 7-hydroxymitragynine, so it's not even present in what we're delivering to these animals. And then we have corianthidine, a little bit below that is from the natural product. There's also speciogynin and painanthine that I'll touch on in just a second that are there, but they disappear very quickly from the plasma. So what is speciosialatin? Speciosialatin is binding at opioid receptors. It's the second most prevalent in the rat model that we've seen. It does have, again, partial agonist effect. It does not recruit beta-arrestin, and it is another opioid molecule within the plant. We've also looked at corianthidine. I mentioned corianthidine is very minor in its effects, but we know that it has high affinity as an alpha-1 adrenergic antagonist. And this is a very interesting piece because it is blocking some of those withdrawal type symptoms or effects. We also looked at painanthine and speciogynin. And what I'm showing you here is that these molecules, which are the second and third most abundant molecules in kratom plant itself, are very high affinity serotonergic ligands. In fact, 5-HT1A agonists have been reported to reverse opioid-induced respiratory depression. And so this is a very interesting piece of the puzzle as well that may add to the safety profile of kratom as a whole. And what you see here between painanthine and speciogynin, the only difference is this double bond that exists outside of the molecule. And the only difference between this molecule and mitragynine is the stereochemistry or the handedness of this molecule. So left versus right hand molecule. Again, high affinity for serotonin receptors and affinity for opioid receptors. So what does this mean in terms of analgesic efficacy? And again, we're looking at these molecules by themselves, but we compared this in an analgesic measure, serotonin-mediated analgesia pathways. And what we could show was that with a selective serotonin antagonist, we could block the analgesic efficacy provided by these molecules. So now we have analgesia that can be created by not only opioid receptors, we have analgesic relief coming from adrenergic receptors as well as serotonergic receptors, what we've seen. And then finally, I'll add to the human clinical trial piece. This was not done by our lab. This was done by Mary Payne's lab at Washington State University College of Pharmacy. And this is some of the pharmacokinetic data that they produced, which is very much in line with the same Cmaxes that were seen in the data that Dr. Hustis presented to us. But just to give you a little bit of different picture, mitragynine was the key major alkaloid within the product that they gave. But what was seen, and they monitored for more than just mitragynine and 7-hydroxymitragynine. And in this case, we see that species sialatine is actually the major alkaloid that becomes exposed in the blood supply. And so this, as we mentioned, has opioid activity and may be more responsible for the analgesic efficacy than any other compound in the plant, at least to our hypotheses at the moment. So this is published in the literature. And then I'll just leave you with, there's been such a long history. We've heard of the safe use in Southeast Asia in the traditional manner. It has been associated with a range of adverse events in the Western world. Alkaloids are definitely structurally different from opioids. They are very different in their pharmacokinetic and pharmacodynamic properties, as I've outlined. I term this as disruptive pharmacology. Nobody really knows where to put this. As scientists, we always want to put it into a bucket. This molecule is an opioid. This molecule is a adrenergic agonist. Whatever we want that sort of clean description. But these are really disruptive in their pharmacology. Products in the US are really not the same as the traditionally utilized fresh leaf tea preparations, and we're still trying to understand what the regulations, if any, should be on alkaloid limits and what recommendations we can put forth. But the take-home overall message is there's still a lot of gaps in the science around this plant. We need a lot more research to take place. We need to do a lot more chronic studies and controlled clinical studies. And I'll leave you with the fact that we strongly believe a solution to, or part of the solution to the opioid epidemic could come from nature, where the opioid epidemic actually originated from itself. So thank you.